In this video, we're going to talk about molecular cloning. Now, molecular cloning allows scientists to isolate and have a renewable source of a piece of DNA from any organism. And it involves taking that piece of DNA that we call it foreign DNA because it's foreign to bacteria, isolating that DNA and putting it into bacteria. And since bacteria are easy and cheap to grow, it gives scientists a cost-effective, renewable way of having unlimited amounts of that piece of DNA. So let's take a look at how it's done and what is required. One of the things that is required is restriction enzymes. So you're familiar with what an enzyme is, and it basically catalyzes a chemical reaction. One type of group of enzymes called restriction enzymes, their substrate is a specific sequence of DNA. So if this is a piece of DNA and it has this sequence GGA TCC, this is a sequence that is specifically required by one particular enzyme. And once that enzyme binds the DNA with this particular sequence, it will cut the backbone of the DNA into places on the two strands. You may be thinking, well, why do these enzymes exist? Well, it's a way of, usually these enzymes are isolated from bacteria. These enzymes is a way for bacteria to protect the bacterium from foreign pieces of DNA that gets into the bacterium. So anyway, scientists leverage these enzymes to do molecular cloning. Usually, but not all, many but not all restriction enzymes cut the backbone on the DNA on uneven places, such that after they have cut it, something called the overhang appears at the ends of the DNA that is cut, which we call sticky ends. Why are they sticky? Well, because here are these bases that have no partners. So now if you cut this piece of DNA with the restriction enzyme and you have these sticky ends, you can have DNA from another source, cut them with the same sequence, within the same sequence, and then they will have complementary sequences. So you can bring a piece of DNA in here and join them together by the virtue of hydrogen bonding. So let's look at the steps that are involved. Now let me bring my pen. So you see in here we have the foreign DNA and we have this thing called a plasmid. What is a plasmid? Well, if you look at a bacterial cell, we said the genome for a bacterial cell on this big circular piece of DNA, which we call the bacterial chromosome. But in bacteria, there are also little pieces of DNA, which we call plasmids. And these plasmids can independently replicate and propagate themselves during the cell division of bacteria. So these are two entirely different pieces of DNA. And often plasmids carry the antibiotic resistance genes that bacteria share with one another. So there are a number of different important regions of a plasmid that you need to learn about. So if we talk about this plasmid, just let's say this is a plasmid, you see a number of things. So you see a gene in here which is labeled AMP, and this is a gene on the bacteria that produces a protein that causes resistance to an antibiotic, ampicillin. And I'll explain the significance of some of these. Just remember that this AMP refers to a gene that produces a product that allows a bacteria carrying the plasmid to be ampicillin resistant, to be resistant to that antibiotic, so it can grow in the presence of antibiotic. So if you have two bacterial cells, one that is carrying the plasmid and one that is not carrying the plasmid. Now, if you grow these in the presence in a medium, in a growth medium that has ampicillin, this one cannot grow, but this one that has the plasmid can grow because it carries that ampicillin resistant gene. It's a way of scientists knowing which bacteria have taken up the plasmid. Then we have one thing in here which is called the restriction site. The restriction site, or sometimes called multi-cloning sites, this is a place where it's riddled. Scientists designed these to be riddled with recognition sequences for all sorts of different enzymes, all sorts of different restriction enzymes. So that's where that foreign piece of DNA is going to be placed. You also notice something in here, the LAC-Z gene. Well, what is that? Let me change the color of my pen to blue. Okay, so the LAC-Z gene, I draw it like this because this multi-cloning site sits in the middle of the LAC-Z gene. So this LAC-Z gene codes for a protein that breaks down a molecule, which when it breaks down the molecule, it turns blue. So let's say you start with a molecule that we, for argument's sake, we say that molecule is not blue. That's its color is not visible. It's colorless, but I can't really draw colorless here, so I use this type of blue. Okay, so now if you treat this molecule with an enzyme, LAC-Z, so you treat it with the LAC-Z gene product, then what happens, this produces a product that is blue. So what's the significance of this? Well, any colony that carries this plasmid is going to be colored blue. So if there is a plasmid that has, if there is a bacteria that has the plasmid, it's going to show up blue when you plate them, when you put them in the presence of a growth medium that has this molecule in it. Now, what happens when you clone a gene in here? What happens to this LAC-Z gene? So if you take this gene and clone it in here, you basically disrupt the gene for LAC-Z. So this LAC-Z isn't going to work anymore. So therefore, the bacteria who have plasmids, bacteria which have picked up plasmids that have a foreign gene in it, they are not going to look blue. So you're going to look, so if you have a combination of bacteria, the ones that have a plasmid that has the insert in it, they're not going to be blue. So you'll see, again, I'll show you as we pass through the slides. Okay. So we have the foreign DNA, we have the plasmid, there is a restriction site, and now we're going to cut the foreign DNA and the plasmid with the same enzyme. And that enzyme produces these sticky ends that we talked about, remember? So this sticky end on the foreign DNA is going to be complementary to the sticky ends on the plasmid. So then when you put them together, these two pieces of DNA can hydrogen bond and be held close. See, they're hydrogen bonding in here. But then the hydrogen bonding is not a covalent bond. So what you need to add, once you mix the two pieces of DNA, you have to add an enzyme called DNA ligase. What DNA ligase does is forms covalent bond between these pieces of DNA that are held together via hydrogen bonding. So now what is the next step in this process? The next step is to make plenty copies of this, what we call recombinant DNA. How do we do that? We do it by a process of transformation. We're going to force bacteria to take up the DNA. Transformation happens when bacteria pick up external DNA. So you expose bacteria to foreign DNA, pieces of DNA, and then they take it up into themselves. It's called the transformation. You have to treat bacteria with a certain kind of solution that make it more likely that these bacteria can pick up the DNA. Now you notice here that some bacteria have not picked up the foreign DNA and some have. That's because the transformation is not a very efficient process. So if, for example, if you have 1 million bacteria and then you treat them with this recombinant DNA, a very small minority of them are going to pick up the DNA. So how are you going to distinguish between the ones that have the plasmid and the ones that don't have the plasmid? This is where those elements that we just talked about come into play. So you're going to take this mixture of DNA and plasmid and then put it on a growth medium. And this growth medium has two things. One is ampicillin. So if there are any cells any bacterial cells that have not taken up the plasmid, they will not be able to grow. So all these cells have a plasmid because the plasmid has the ampicillin resistant gene. Now you notice there are different colors in here, and that brings us to that LAC-Z gene that we talked about. So what is the purpose of the LAC-Z gene? I explained it earlier, but I want to point out again that sometimes when you cut a plasmid with an enzyme, sometimes this plasmid snaps back. So instead of getting in this foreign DNA into it, it just snaps back, it gets close. So some of these bacteria actually have a plasmid that has none of these inserts in it. So if you remember, we said if the plasmid has no insert in it, it makes the product of the LAC-Z gene, and therefore in the medium where you have the substrate for the LAC-Z gene, it's that enzyme is going to break the substrate and make the bacteria blue. So here it is. The ones that are blue, they do not have a foreign gene in the plasmid. But if you insert the foreign gene into the plasmid, you basically destroy this LAC-Z gene, and there's not going to be any product, a blue product made. So the colonies that are white have plasmids that have insert in them. And by insert, I mean in these pieces of DNA. So what you want on these plates, you want these cells that are white and the cells that grow in the presence of ampicillin. So that's how you find the clones. Now each of these circles actually is not it's just one cell. It's about 10 to the power of about 1 million cells that all came from one cell. So each of these cells, when you put them on the growth medium, they're going to keep dividing and dividing and give you this visible circles that are either white or blue. And since each of these 10 to the power of 6 cells came from one cell, we call them a clone. And clone, the literal meaning if you've heard of it, is meaning things that are identical. And because these cells divide by asexual reproduction, all the cells are identical and all of them have either plasmid with foreign gene in it or they have plasmid with no foreign gene in it.